Welcome to Key to My Research, the science podcast by the Excellence Strategy at the University of Tübingen. In this podcast, we explain in a simple way how outstanding scientists are researching complex topics that affect our everyday lives. My name is Jennifer and I'm your host. In today's episode, we get to know Professor Hannes Link.
We can also address climate change by reprogramming bacteria, for example, such that they fixate CO2.
He's a professor for microbiology and bacterial metabolomics at the University of Tübingen. And he's an expert when it comes to the metabolism of bacteria.
The key to my research is to stay motivated, that I have questions that I think are interesting enough, that I spend a lot of time thinking about them, and then I can develop, again, interesting ideas. And then maybe the key is just to select the right idea out of these many ideas that you have.
Professor Link's passion for DNA and bacteria began early on when he was still a student.
One key moment was during my studies when I learned about how DNA is translated to RNA, how this is translated to protein, so how the genetic code is read and how this genetic code, somehow, how function emerges from the genetic code. This was really fascinating. I was actually a bit angry that I never learned this at school. So this is a really key moment which motivated me to work on that topic, how DNA encodes all the functions for life.
Today, Professor Link is trying to really understand the metabolism of bacteria. He and his teams focus on 3 areas. Before we dive into these 3 areas, let's try to understand why metabolism is so important.
If you understand metabolism, then you immediately have a wide range of applications. And this could be microbiomes, where we investigate metabolism in the microbiome, or the metabolism-antibiotic interaction, or synthetic biology, where we try to reprogram metabolism. And our role, often these are collaborations, our role is really to investigate metabolism and to modify metabolism.
So this is why metabolism is so important for Professor Link and his teams. But before one can understand the metabolism of bacteria, one has to understand the bacterium itself. And this is fascinating because bacteria are in some ways very simple and at the same time very complex.
Bacteria, in order to grow, they have to produce building blocks. And out of these building blocks, they produce then all the components that they need to build a new bacterial cell. And bacteria can do something special. This is something we humans cannot do. They can make these building blocks from very simple nutrients. They can take a simple sugar molecule or something even simpler, something like CO2. And out from these simple molecules, they build all the building blocks they need to grow. Sometimes I use the analogy of a factory, like a car factory, but here the factory is building a bacterial cell. So this is the end product. And this factory starts with a simple starting material like a sugar or like something like CO2. And then there are hundreds of individual steps. And in these steps, the cell is building a new bacterial cell as the final end product. And the nice thing is we know each of these individual steps very well. We know what they're doing, where they are in this bacterial factory. But what we don't know is how this is controlled, how all of this functions together.
And this is what Professor Link and his teams are trying to find out in their 3 main areas of research. One of them, as mentioned, is the microbiome. The microbiome is becoming more and more important. And microbiomes can be found in our bodies, but also in plants, as you may have heard in one of our previous episodes with Professor Kemmen. One of the best-known microbiomes is in our guts. But Professor Link and his teams are taking a closer look at another microbiome.
We started now with the nasal microbiome. So in Tübingen, I have many colleagues working on microbiomes. We have Lisa Meyer, she works on the gut microbiome, and this is all very interesting. But since we're working with E. coli and already know how complex a single bacterium can be, it was a bit daunting to me Now suddenly you don't have a single bacterial factory, but you have many of them and they're interacting. That's why we decided for the nasal microbiome. So it's a little smaller microbiome where not so many bacteria are living together. So we started now analyzing the nasal microbiome. And again, we're focusing on metabolism. We're asking what happens now if a bacterium doesn't work in isolation. So this metabolic factory is not alone. It has now 10 other bacteria. And from all we see at the moment is that they're highly interacting. So these factories kind of work together, they share some nutrients and maybe also develop some new functions.
The microbiome, like the one found in our guts, can be influenced by what we eat. It can also change over time. But the nasal microbiome is different.
You don't have so much ways to modulate the nasal microbiome. And this is, for example, something we would like to find out. How can we remove a major pathogen from the nose? So 1/3 of humans, they have Staph aureus in the nose. This is this pathogen MRSA, which causes a lot of problems in the clinic. And often if you go to the clinic, you get a cream which you can smear into the nose that Staph aureus is removed from the nose. And there we would like to find out alternative therapies or alternative ways of somehow to modulate the nasal microbiome such that Staph aureus can be removed. And one way could be, for example, probiotics, that you just put another bacterial strain into your nose and then you have a more healthy nasal microbiome.
Professor Link's second area of focus comprises metabolism and antibiotic interaction.
So we're investigating antibiotics, so how antibiotics work and how antibiotics kill bacteria, because the antibiotic in the first place, it just inhibits the bacterial growth, so the bacterium cannot grow anymore. But what we actually want is that the bacterium dies, that it really disappears from our body. And there we're investigating a little bit the mechanism that lead to bacterial cell death. And there we learned a little bit that it seems that metabolism plays an important role, because if bacteria find a way to slow down their metabolism, then they can somehow prevent that they die from an antibiotic. And this can be dangerous in the clinic, or if you have an infection, if you get an antibiotic, you take it for several days, the bacteria will not grow, but then actually once you remove the antibiotic from your body, then they can grow again because they're not died. So you really have to clear the bacteria or the pathogens from your body. And this is why we would like to find out, how can we improve this killing? For example, is there a certain diet that you could take with an antibiotic such that the antibiotic is more effective?
Before we look at the third main area of research, synthetic biology, we need to understand the methods used to do the research. A very important method is CRISPR. This is a protein that allows scientists to actually cut DNA. And this has changed a lot, especially in biology.
CRISPR, it's called an endonuclease. It's a protein, Cas9 for example. And this protein can cut DNA. And it can cut DNA precisely at the position where I can program it. So it's a programmable scissors. So I can cut the DNA exactly at the point where I like to do this. And at this point, I can then insert a new gene or a mutation, or I can delete something. So it really allows us now to reprogram the genetic code of a cell.
Now let's focus on synthetic biology, or genetic engineering. This is Professor Link's third main area of research. But in order to do synthetic biology, you first need what is called systems biology.
In systems biology, we try to understand how these bacteria are working. So that we try to understand how are the molecular components inside the bacteria, how they're interacting, and which networks emerge from these interactions. Often it's very messy, these are very complex networks, but these networks really allow the cell to self-organize, to self-repair, and to function in a way how they do. So this is the task of systems biology, that we map out these networks, that we understand how they function, these networks, and then we can use this understanding for synthetic biology. And synthetic biology is really all about genetic engineering, about writing the genetic code or coding with DNA. So synthetic biology has many applications, really a wide range. We all know the mRNA vaccines that we use now for the coronaviruses, but we can also address climate change by reprogramming bacteria, for example, such that they fixate CO2. This is something we do, for example, in a collaboration with the lab of Ron Milo in Israel. So what we do there, we can transplant the CO2 fixation pathway from a plant into a bacterium like E. coli. So E. coli naturally just uses the sugars or something that you can use in our gut. But now we can just transfer a pathway from a plant, let's say, into E. coli and then try to get this functioning such that E. coli is not eating sugars anymore, but that it's eating CO2. And then you can imagine then you can address a lot of problems if you can make chemicals out of CO2. Synthetic biology, I said it's engineering biology, but we're not there yet. So I'm a chemical engineer, so I learned how to build a chemical plant. And this works usually, we design this at the computer, and then we build the plant and we know the plant is working. And this is something we cannot do with a biological system. Let's take the CO2 fixation pathway. In our case, we're not there that we design this at the computer, we install this in E. coli, and it's working. It's still a lot of trial and error. And our CRISPR allows us to do a lot of trial and error, to do this really at a massively large scale. So we're trying at the moment to make hundreds, thousands of genetic changes and then just try which of them is helping E. coli in this case to work with this CO2 fixation pathway. And from this trial and error, we can then kind of iterate a little bit and get some more understanding and then do this in a more rational way.
At the University of Tübingen, Professor Link is part of the Controlling Microbes to Fight Infections CMFI Cluster of Excellence. This is a great enrichment for his research.
So in the cluster, the important thing is that we have experts in these different research areas, infection biology, and especially the microbiome, because it's a very dynamic, it's a very complex and competitive field or fields. So doing this on your own is very demanding. So often these are collaborations where we work, for example, with colleagues on the gut microbiome, and then we can take our methods, work with these colleagues which have other expertise, for example, in these different bacteria. We are focusing a lot on E. coli because it's simple, it's well known. And then moving already to another bacterium is somewhat difficult, and therefore it needs collaborators. And this helps a lot in Tübingen.
This was Key to My Research, a podcast produced by Changing Time in cooperation with the University of Tübingen. For more information and the links to the sources, see the show notes below. There, you can also find a link to the whole interview with Professor Link. If you enjoyed this episode, make sure to subscribe to the podcast, leave a review, and recommend it to your friends and colleagues. Authors: Chris Veit and Jotti Fotiadis. Special thanks to Professor Hannes Link, Heiko Heil, Oliver Häusler, Kurt Schneider, and Oliver Lichtwald. My name is Jennifer. Thank you for listening and see you next time.